Today, we're going to talk about a real game changer in the treatment of metastatic colorectal cancer, Fruquintinib. And this is so much more than just a new drug. It represents a fundamental shift, one that's deeply rooted in our understanding of the molecular, genetic, and epigenetic drivers of this disease. We're going to dive deep into the science that's changing the way we fight this cancer.
You know, this quote really cuts to the core of our challenge: metastasis. For our patients, it's the most devastating part of the diagnosis. But what we've come to understand is that it's not some chaotic, random event. It's a highly regulated process. There's a playbook, and that playbook is written at the genetic and epigenetic level. This is the battlefield, and it's precisely where a targeted therapy like Fruquintinib has to make it stand. For far too long in metastatic colorectal cancer, we've hit this therapeutic ceiling. After first and second-line therapies, our options get very limited very quickly. You see the overall survival numbers start to plateau right around 30 months. For those patients with refractory disease, we've been in desperate need of effective, well-tolerated options. This is the critical gap that Fruquintinib was designed to address.
Okay, so here's our game plan. We'll start with the clinical evidence, the hard data that validates this drug. Then, we're going to pop the hood and look at what makes it work so well, its incredible selectivity. From there, we'll get into the most exciting part, the precision layer, exploring the genetics and epigenetics that dictate response. And finally, we'll look ahead to the future of truly personalized therapy.
So let's start with the proof. Because in our field, a novel mechanism is just a theory until it's backed by robust clinical data. For Fruquintinib, that validation came from two landmark, global phase 3 trials. The first was the FRESCO trial out of China. And the results were frankly impressive. It demonstrated a median overall survival of 9.3 months compared to just 6.6 months for placebo. Now, what does that actually mean for our patients? It means a 35% relative reduction in the risk of death. In a heavily pretreated population, that is a significant and clinically meaningful benefit. Then came FRESCO 2. This was a global study with an even more challenging, heavily pretreated population. And the results held. 7.4 months versus 4.8 months. But here's the kicker. The benefit was consistent even in patients who had already been treated with other anti-VEGF therapies. This solidified Fruquintinib's role as a new global standard of care for these patients. Okay, so the clinical data is clear, but why does it work so well? And why is it so well tolerated? The answer is all about the power of its very specific, very selective mechanism. At its core, Fruquintinib is an oral tyrosine kinase inhibitor. But it's not just any TKI, it's a highly selective one. It zeroes in on the vascular endothelial growth factor receptors, VEGFR 1, 2, and 3. Tumors desperately need these receptors to build new blood vessels, a process we call angiogenesis. Fruquintinib works by binding inside the kinase domain, right where the energy molecule ATP is supposed to go. By blocking ATP, it essentially pulls the plug on the receptor's power source. No power, no signal, and the whole process shuts down. And this right here is the secret sauce: selectivity. Think of it like this. Many other inhibitors are like a sledgehammer. They hit VEGFR, sure, but they also hit a whole host of other kinases like PDGFR, FGR, KIT, you name it. That leads to a lot of collateral damage, what we call off-target toxicities. Fruquintinib, on the other hand, is a precision-engineered key, designed for one very specific set of locks: the VEGFRs. This remarkable precision is why we see a much more manageable side effect profile, which allows our patients to stay on therapy longer. And you can see the powerful domino effect this has. By blocking that single phosphorylation event at the receptor, Fruquintinib brings the entire downstream signaling cascade to a grinding halt. We're talking about major pro-survival pathways like RAS, MEK, ERK, and PI3K, AKT-mTOR. They're all shut down. The end result? The endothelial cells can't proliferate, they can't survive, and they can't migrate. The tumor is effectively starved of the blood supply it needs to grow and metastasize. So now we understand how it works, but that brings us to the next, arguably more critical question. Why does one patient respond so well while another doesn't? To answer that, we have to look beyond the drug itself and into the patient's own biology. We have to peel back the precision layer of genetics and epigenetics. It all starts with the germline DNA, the code we inherit. Small variations or polymorphisms can have a huge impact on therapeutic response. Variations in the target genes themselves, like VEGFR2 or 3, can change how tightly the drug binds.
Variations in ligand genes like VEGF-A can determine how much of the target is even there to begin with. And then, crucially, there are variants in the genes that handle the drug itself, like CYP3A4 for metabolism and ABCB1 for transport, which dictate how much active drug is available to do its job.
But it doesn't stop with our fixed DNA sequence. There's this whole other layer of regulation called epigenetics. You can think of these as chemical sticky notes that are placed on our DNA, telling genes when to turn on and off. Things like DNA methylation can silence key tumor suppressors, while histone modifications can ramp up the expression of VEGF. The critical point here is that, unlike the genetic code, the epigenome is dynamic. It can change, and it's often how tumors develop resistance, but this also means it presents a whole new class of potential therapeutic targets.
So, what do we do with all this incredible molecular information? This understanding is what allows us to truly look forward, to move beyond a one-size-fits-all approach and toward the future of genuine precision oncology.
And that really is the million-dollar question, isn't it? We have this wealth of molecular knowledge accumulating at an incredible pace. So how do we effectively translate it from the research lab to the clinic to make better, more informed decisions for the individual patient sitting right in front of us?
The answer lies in the hunt for predictive biomarkers. We're casting a wide net, looking for reliable signals everywhere. In the blood, through liquid biopsies, looking at things like circulating VEGF levels or exosomal microRNAs. In the tumor tissue itself, analyzing gene expression signatures and mutation status. We're even using advanced functional imaging to see, in real time, how the drug is impacting the tumor microenvironment. The goal is to integrate these different data streams, a true multi-omics approach, to build a complete molecular dossier for every patient's cancer.
And once we have that molecular dossier, it opens the door to the next frontier: rational, intelligent combination strategies. This isn't just about throwing drugs at a tumor to see what sticks. This is about precision. By profiling a tumor's specific vulnerabilities, we can pair Fruquintinib with its ideal partner. For a RAS wild-type tumor, maybe that's an EGFR inhibitor. For an immunologically cold tumor, Fruquintinib can help normalize the tumor vasculature, making it more visible to the immune system and ripe for combination with immunotherapy. We can even think about using epigenetic drugs to reverse acquired resistance. It's about designing a bespoke therapeutic strategy for each individual cancer.
And that brings us to the ultimate conclusion. We are truly on the cusp of a new era in oncology. It's an era where therapy will no longer be defined simply by histology or location in the body, but by the unique molecular signature of the cancer itself. Understanding this deep, intricate interplay between genetics, epigenetics, and targeted therapies like Fruquintinib isn't just an academic exercise, it is the very blueprint for the future of personalized cancer medicine.